Have you ever wondered how we can look beyond genomics to gain deeper insights into biology? With advancements in proteomics, scientists can now unlock a whole new layer of understanding, helping to discover biomarkers, unravel disease mechanisms, and identify novel therapeutic targets. But how can we integrate this technology into our research? Today, we're joined by Dr. Cindy Lawley, Senior Director of Global Population Health at Olink Proteomics, part of Thermo Fisher Scientific. With a wealth of experience in developing solutions to understand genetic risk in diverse populations, Cindy has led numerous initiatives that have helped push the boundaries of proteomics research.
I direct population health at Olink, part of Thermo Fisher Scientific. Directing population health means I have the privilege of working with scientists leveraging next-generation sequencing proteomics. This is what I'll talk about today. My name is Cindy Lawley. I was invited to contribute to Teach Me in 10 to talk about how researchers, primarily geneticists, are using NGS proteomics. So let's start. But let's start by talking about genetic data. So technology breakthroughs have changed how we collect genetic data, and this has been happening over the last 20 years. Today, it's easier and cheaper than ever before to compare my genetic differences to your genetic differences. These differences form the foundation of precision medicine, also called individualized medicine. So that's because we vary at a genetic level. The promise of being able to treat us each as individuals in healthcare has its foundation in understanding how we differ genetically. It's also proteomic, as I'll show you. So when I say genetic in this image, I'm talking about DNA. So generally, DNA works from the inside of the cell nucleus, illustrated here, and it gives instructions that are carried out by the RNA. So what are those instructions? Well, those instructions are typically about making proteins. So to fully understand how we differ genetically, back to my point about individualized medicine, we need to know how we differ from DNA to RNA and to proteins. So maybe you process sugars more efficiently than I do. Maybe your body takes them out of your bloodstream more easily and is able to get them into your muscles and use them more quickly. If that's the case, perhaps I have a higher risk for type 2 diabetes. That's about the function of proteins in your body as a result of differences that are coded mostly genetically. So let's talk about the jobs of proteins. Proteins are doing the work of the cell by carrying out the instructions coming from DNA. So measuring proteins would be very useful in this world. And just like advances in collecting genetic information, we've seen advances in collecting proteomic information. So looking broadly across lots of proteins in lots of samples is where NGS proteomics comes in. And only recently have we seen ways to measure proteins in large enough populations that we can see what proteins can add to our understanding of how we differ as individuals. So how do we measure proteins in lots of samples in large populations? Well, one way we do this is to use antibodies. In this video, you'll see the antibodies are these Y-shaped structures. And this is how Olink does this. So this is Olink's proximity extension assay. We call it the PEA assay. We use 2 antibodies to find each protein, and attached to those antibodies are these little single-stranded tags of DNA. And they're attached to those antibodies so that they can find each other once the antibodies find the right protein. Once they find each other and they hybridize, they can be extended, as you've seen here in this tag. And then that tag can be counted on a next generation sequencing instrument, one of the ways that we've advanced our ability to collect genetic information. So essentially, we're turning a complex protein signal into a DNA signal, and the counts of those tags reflect the original concentration or amount of the protein in the original sample. Those little tags allow us to track back to protein. They allow us to track back to the sample and read them all out at the same time. So being able to collect this information, like I said, in large numbers of samples is really changing. And one of the ways that it's being manifested and used broadly by researchers is as part of what's called the UK Biobank Pharma Proteomics Project. This is a full cohort study of 600,000 samples. So the UK Biobank is a set of samples of participants that were enrolled, and they started enrollment in the early 2000s. Many of those participants in the UK Biobank, where their samples are being stored, have gone on to develop diseases over time. It's been almost 20 years since then. So genetics have been collected on all participants, so we know how these participants vary genetically. And now 14 pharma and biotech partners represented by this slide are coming together to collect proteins on these same samples so that we'll have genetic information, protein information, and health outcome information on those same samples. Like I said, this is called the UK Biobank Pharma Proteomics Project Full Cohort Study, and represents 600,000 samples. This effort's being led by Chris Whelan. He's from Johnson & Johnson. He's shown here in the lower center figure. So why are these pharma and biotech partners so committed to incorporating proteomics in so many samples where they've already got genetic data? Well, I'll talk a little bit about that, but this 600,000 sample project is being committed because of how much they saw in a pilot study, also using UK Biobank samples. So pharma partners came together and published on a pilot study of over 54,000 samples, and these came out in the October 2023 issue of Nature. What we call the flagship paper that included most of these pharma partner authors. This currently today has over 149,000 accesses. That means that's how many times people have accessed this paper. And it's been referenced around 600 times, often because folks are leveraging these data that are publicly available. And so it's being queried by researchers all over the world, and publications are coming out at a very fast pace. The UK Biobank, with all of its data, is absolutely an international treasure that is founded in the UK, United Kingdom. So now I'll talk about a few of the studies that reference that flagship paper and use the data from that pilot in the UK Biobank pilot study. Again, 54,000 individuals. So the first publication that I'm illustrating here is from a team at Optima Partners. It includes Ben Sun, who is at BMS now, Bristol Myers Squibb. And they built protein scores for 23 different diseases. So what does this mean? These are scores, including proteins that are important in those diseases. And these were for individuals who had had blood collected back in 2006, around 2006. And those individuals went on to develop the same disease. So for 23 different diseases. Because we know who developed the diseases, we could model the proteins and the genetics on how each performed, genetics versus proteomics, in predicting the risk of those individuals getting that disease. And that's typically they're looking at just the next 10 years. So that's the title on this slide. So protein scores for predicting risk of getting a disease in the next 10 years. So let's just use type 2 diabetes as an example, which we mentioned earlier. They found that protein scores could outperform genetic risk scores, so what we learned from genetics, by over 20 points in predictive power. So that's 20 points in a scale of 0 to 100% perfect predictive power. This shows that proteins are improving risk prediction dramatically over genetics alone. And it's important to include proteins in the future, and we expect that to happen. Similar work was led by Karen Papier. This is the team at Ruth Travis's group, and they looked at 19 blood and solid tumor cancers, and they used protein signatures to predict cancer a median of 12 years prior to clinical diagnosis in hospital records from the UK Biobank. And finally, Julia Carrasco-Zanini led work from Claudia Langenberg, looking at over 200 common and rare diseases that were represented in this data set, the UK Biobank. They found for 67 of these diseases, a protein score developed with just 5 to 20 proteins was able to outperform anything a doctor has available to them today for predicting who's going to go on to get that disease, any of these 67 diseases. So NGS proteomics are punching well above their weight in bridging our understanding of how to move from observation to actionable insights, insights that were able to actually move people's healthcare needle. So this is super exciting. This is research use only. Now I'd like to talk about a clinical example that is being built out by Octave Biosciences using multi-protein signatures. So Octave Biosciences and many other groups are building disease-focused or application, need-focused signatures of proteins to take to the clinic. So these are, not tools that are off the shelf at Olink, but these are tools that are being built around proteins where the discoveries have been made on discovery tools. So this slide shows the timeline. They did their first discovery study in 2017. They demonstrated analytical validation in 2020. And within 3 years, they were then able to clinically validate a signature of 18 proteins as part of a dashboard for monitoring individuals with multiple sclerosis who are often diagnosed quite early and quite young in life. This helps monitor their therapeutic journey. Often they need to go from one medication to another medication as they are being monitored for that disease. This MS test, this multiple sclerosis test, is now being accessed in a clinical LDT by over 100 different labs. So this is a remarkable tool. It's part of that dashboard. And again, this is really about individualized medicine, treating individuals based upon their differences and where they're at, here in this case, in their therapeutic journey. So that concludes my Teach Me in 10. I just want to highlight the main points that genetics and proteomics are the foundation to enable individualized medicine, that NGS proteomics is being shown to improve disease risk prediction across many different diseases, and multi-protein signatures are showing themselves to be useful for monitoring disease progression and treatment response in the clinic to take care of patients and improve their patient experience. Thank you so much for joining me for this Teach Me in 10. Feel free to reach out if you have any questions. Thank you.
This episode is brought to you by Olink Proteomics, part of Thermo Fisher Scientific. Olink is redefining the future of multiomics with an innovative NGS-based proteomics solutions. Powered by the proprietary proximity extension assay technology, Olink enables high-throughput, sensitive, and precise protein quantification using next-generation sequencing. Thank you for watching, and see you next time for another Teach Me in 10.